7:42 a.m. June 3, 1987. Robert Henshaw was standing in the southeast corner of a 640-acre wheat field outside Goodland, Kansas, when he noticed the crop along the access road looked thinner than the rest. The wheat was 18 inches shorter there, thinner stalks, lighter color. He had followed every USDA recommendation for atrazine application for 6 years straight. Henshaw had applied exactly 2.2 pounds of active ingredient per acre every spring since 1982. That was the published rate, the recommended rate, the safe rate according to every extension bulletin the government had printed since 1965. By harvest that year, Henshaw would lose $14,600 on that field. The soil tests would show something nobody at USDA had warned him about. And the rate that destroyed his field was printed in official guidelines that would not be corrected until 1987, 22 years after the original recommendation was issued. Before we go further, if this kind of agricultural history matters to you, hit subscribe. These stories disappear when nobody documents them. Atrazine became the most widely used herbicide in American agriculture in 1959. J.G. Chemical Corporation introduced it as a pre-emergence and early post-emergence control for broadleaf weeds and grassy weeds in corn. The chemical worked by inhibiting photosynthesis in target plants. The company promised season-long control with a single application. It promised labor savings over mechanical cultivation. It promised yield increases of 12 to 18% in trials. By 1965, J.G. had sold atrazine to farmers working 26 million acres of American farmland. That year, the United States Department of Agriculture published its first official application rate recommendation, 2 to 2.5 pounds of active ingredient per acre for most soil types. The bulletin was printed by the Agricultural Research Service and distributed through every county extension office in the Midwest. The rate appeared in land-grant university guides. It appeared in commercial applicator training manuals. It appeared on product labels. It became the standard. What the USDA did not mention in that 1965 bulletin was what atrazine did to soil biology at that rate. The research had not been conducted. The testing protocols used to establish the recommended rate measured only two things: weed control efficacy and crop injury visible within 90 days of application. Nobody had tested what happened to the microbial populations in the soil. Nobody had measured what happened to nitrogen-fixing bacteria. Nobody had tracked earthworm populations over multiple seasons. The assumption built into every application rate guideline was that the soil was an inert medium. The chemical killed weeds, the crop grew, what happened in between did not matter because it was not measured. The first documented report of unexplained yield loss in fields treated at USDA recommended atrazine rates appeared in a 1969 agronomic journal published by the University of Illinois. The article was written by Dr. Harold Swanson, an extension agronomist in Champaign County. Swanson had documented 12 cornfields across 3 townships where farmers had followed recommended atrazine rates for 3 consecutive years. In year 3, every field showed an average yield reduction of 14% compared to untreated check strips. Soil nitrogen levels were lower. Organic matter decomposition rates were slower. Earthworm counts were down by 63%. Swanson wrote that the yield loss could not be explained by weed competition because weed control had been excellent in every field. He suggested that repeated applications at the recommended rate might be affecting soil biological activity. The article appeared on page 287 of a journal that county agents did not routinely read. USDA did not revise the recommended rate. Extension bulletins continued to list 2 to 2.5 pounds per acre as standard practice. By 1973, similar reports were appearing in Nebraska, Iowa, and Kansas. Dr. Paul Jorgensen at the University of Nebraska documented 18 fields where continuous atrazine use at recommended rates had resulted in measurable soil compaction, reduced infiltration rates, and declining organic matter despite consistent manure applications. He described the soil structure as deteriorating. He measured a 41% reduction in microbial respiration compared to untreated controls. He noted that farmers were compensating for lower yields by increasing fertilizer applications, which temporarily masked the underlying problem. Jorgensen submitted his findings to USDA in the form of a research summary in February 1974. He recommended that the application rate be reduced to 1.5 pounds per acre and that guidelines include a warning about long-term soil impacts. USDA did not respond. The recommended rate remained unchanged. The mechanism of damage was not mysterious. It was straightforward once anyone looked. Atrazine at 2 to 2.5 pounds per acre did not degrade quickly in most soils. The half-life in clay loam soils ranged from 40 to 60 days depending on temperature and moisture. That meant that in a typical growing season, significant atrazine residue remained in the root zone long after the target weeds were dead. The chemical bound to organic matter in the soil. It concentrated in the top 6 inches where the highest populations of beneficial bacteria, fungi, and earthworms lived. At concentrations above 1.8 parts per million in soil solution, atrazine inhibited nitrifying bacteria. Those bacteria converted ammonium to nitrate, the form of nitrogen that corn could use. When nitrifying bacteria populations dropped, nitrogen availability dropped. Farmers saw the symptom, which was lighter green corn and lower yields. They applied more nitrogen fertilizer. The fertilizer provided short-term relief but did not address the underlying cause, which was that the biological system that processed nitrogen in the soil was being suppressed. Atrazine also affected mycorrhizal fungi. Those fungi colonized corn roots and extended the effective root surface area by a factor of 10 to 100 depending on soil type. The fungi increased phosphorus uptake, improved drought tolerance, and contributed to soil aggregation. Research published in 1976 by Dr. Elaine Caldwell at Michigan State University showed that mycorrhizal colonization rates in corn roots dropped by 52% in soils treated with atrazine at 2 pounds per acre compared to untreated soils. Caldwell measured phosphorus uptake in greenhouse trials. Corn grown in atrazine-treated soil took up 38% less phosphorus, even when soil phosphorus levels were identical to controls. The fungal network that delivered phosphorus to the roots had been damaged. Caldwell published her findings in the journal Soil Biology and Biochemistry. She concluded that the USDA recommended rate was high enough to disrupt beneficial fungal relationships that were critical to nutrient uptake. She recommended that application rates be reduced or that guidelines include warnings about mycorrhizal suppression. USDA did not revise the guidelines. Earthworm populations declined wherever atrazine was used at recommended rates for more than 2 years. Research conducted by Dr. Thomas Blevins at Ohio State University between 1974 and 1978 documented earthworm counts in 140 fields across 8 counties in central Ohio. Fields treated with atrazine at 2 to 2.5 pounds per acre for 3 consecutive years showed an average earthworm population of 43 individuals per square meter. Untreated fields averaged 176 individuals per square meter. The decline was consistent across soil types. Blevins noted that earthworm activity contributed to soil aeration, water infiltration, and organic matter distribution. He measured water infiltration rates in treated and untreated fields. Fields with depleted earthworm populations had infiltration rates 51% lower than untreated fields. That meant more runoff, more erosion, and more standing water after heavy rain. Blevins submitted his findings to the USDA Soil Conservation Service in 1979. He recommended that atrazine guidelines include a discussion of earthworm impacts and that farmers be advised to reduce rates in soils with naturally low earthworm populations. The recommendation was noted in internal correspondence but did not result in a revision to published guidelines. By 1980, the pattern was documented in enough locations that it could no longer be dismissed as regional variation or anomaly. Studies from Illinois, Nebraska, Iowa, Kansas, Ohio, Michigan, Indiana, and Missouri all reported similar findings. Atrazine at USDA recommended rates suppressed soil biological activity. The suppression was measurable. It was repeatable. It resulted in declining yields over time despite increasing fertilizer inputs. The solution was obvious: reduce the recommended rate, but USDA did not reduce the rate. The 1980 edition of the Extension Bulletin on Atrazine use in corn listed the same application rate that had been published in 1965, 2 to 2.5 pounds of active ingredient per acre. The institutional resistance to revising the rate had 3 sources. First, JG and later CBJG, which acquired the atrazine product line, had conducted the original efficacy trials that supported the 2 to 2.5 pound rate. Those trials measured weed control and acute crop injury. They did not measure soil biology. JG had no financial incentive to fund research that might result in lower application rates and therefore lower sales. Second, USDA agronomists who had authored the original guidelines were reluctant to revise recommendations that had been in place for 15 years. Revising the rate would require acknowledging that the original rate had been based on incomplete testing. Third, the economic incentive for farmers was mixed. Lower application rates meant lower herbicide costs, but they also meant risking incomplete weed control, which could result in catastrophic yield loss. Farmers who had been using 2.2 pounds per acre for a decade were not eager to experiment with 1.5 pounds per acre unless they had strong evidence that the lower rate would still control weeds. The person who finally forced USDA to address the issue was not a researcher, it was a farmer. Robert Henshaw had purchased 640 acres near Goodland, Kansas in 1981. He paid $167,000 for the land. He financed $120,000 of that through Farmers Home Administration. His first crop was wheat in 1982. He followed every USDA recommendation. He applied atrazine at 2.2 pounds per acre that spring based on the Extension Service Bulletin. Weed control was excellent, yield was strong. He did the same in 1983, 1984, 1985, and 1986. By the spring of 1987, Henshaw began noticing that the wheat along his access roads and in the areas where he turned the sprayer looked weaker. The stands were thinner, the color was off. He assumed the issue was compaction or uneven fertilizer application, but soil tests showed adequate nitrogen and phosphorus. The compaction was no worse than the rest of the field. In June 1987, Henshaw walked the field with an extension agent named Dennis Cole. Cole took soil samples from the weak areas and from areas where the crop looked normal. He sent the samples to the Kansas State University Soil Testing Lab. The results came back 3 weeks later. The weak areas had atrazine residue concentrations of 2.4 parts per million in the soil solution. The normal areas had concentrations of 0.9 parts per million. The weak areas also had microbial respiration rates 60% lower than the normal areas. Cole called the KSU agronomy department. He spoke to Dr. Laura Pfeiffer, a soil microbiologist who had been studying atrazine impacts since 1983. Pfeiffer told Cole that atrazine at concentrations above 1.8 parts per million suppressed beneficial soil bacteria, and that repeated applications at USDA recommended rates were likely building up residue levels that damaged soil biology. She told him that the recommended rate had been based on weed control trials that did not measure soil biological impacts. She told him she had submitted research summaries to USDA in 1984 and 1986 recommending a rate reduction. She told him the recommendations had been ignored. Henshaw lost $14,600 on the 1987 wheat crop. The weak areas yielded 18 bushels per acre instead of 42. He had applied atrazine exactly as USDA had recommended. He had followed the guidelines, and the guidelines had destroyed the biological health of his soil. Henshaw was not interested in filing a complaint or writing letters. He was interested in not losing money. In August 1987, he drove to the USDA Agricultural Research Service office in Manhattan, Kansas. He brought his soil test results. He brought his application records. He brought his yield maps. He asked to speak to the regional director. The director was not available. Henshaw left the documents with a secretary and said he would be back in 1 week. He came back the following Thursday. The regional director, a man named Carl Brennan, met with him for 35 minutes. Brennan reviewed the soil tests. He reviewed the application records. He told Henshaw that the recommended rate was based on the best available science at the time it was published. Henshaw asked when that was. Brennan said 1965. Henshaw asked if any research had been conducted on soil biological impacts since 1965. Brennan said he would look into it. 3 months later, in November 1987, USDA published a revised bulletin on atrazine use in corn and wheat. The new recommended rate was 1.5 to 1.8 pounds of active ingredient per acre for most soil types. The bulletin included a section on soil biological considerations. It noted that repeated applications at rates above 1.8 pounds per acre could suppress beneficial bacteria and fungi, and that farmers should monitor soil health indicators including earthworm populations and organic matter levels. The revision was not announced with a press release. It was not accompanied by an explanation of why the original rate had been too high. It was simply published and distributed through county extension offices. The 1965 rate had been in use for 22 years. During that time, millions of acres had been treated at rates high enough to damage soil biology. Nobody at USDA acknowledged the oversight. Nobody explained why the original guidelines had not included soil biological testing. Nobody contacted farmers who had been following the recommendations. The financial cost to farmers who followed the original guidelines is difficult to calculate precisely, but estimates exist. Research published in 1990 by agricultural economists at Iowa State University estimated that corn and wheat yields on fields treated with atrazine at rates above 2 pounds per acre for more than 3 consecutive years were, on average, 11% lower than yields on fields treated at 1.5 pounds per acre or lower. In 1987, approximately 68 million acres of American farmland were treated with atrazine. If half of those acres had been treated at the higher rate for 3 or more years, and if the average yield loss was 11%, the total production loss in 1987 alone would have been equivalent to $890 million in lost revenue at 1987 crop prices. That does not include the cost of additional fertilizer applications farmers used to compensate for declining soil productivity. It does not include the cost of soil remediation. It does not include the value of lost earthworm populations or degraded soil structure. The person who documented the full extent of the soil biological damage most systematically was Dr. Marion Poston, a microbiologist at the USDA Agricultural Research Service Lab in Beltsville, Maryland. Poston was assigned in 1988 to conduct a comprehensive review of atrazine soil impacts. She reviewed 147 published studies. She conducted her own field trials on research farms in Maryland, Iowa, and Kansas. She measured microbial populations, enzyme activity, nitrogen cycling rates, and earthworm counts in soils treated at various atrazine rates over multiple years. Her findings were published in 1991 in a USDA technical bulletin that ran to 93 pages. Poston concluded that atrazine at rates above 1.7 pounds per acre caused measurable suppression of nitrifying bacteria, mycorrhizal fungi, and earthworm populations in most soil types. She found that the suppression was cumulative. Soils treated at 2.2 pounds per acre for 5 consecutive years had microbial activity levels 58% lower than untreated soils. She found that it took an average of 4 years for microbial populations to recover after atrazine applications were discontinued. She noted that the original USDA recommended rate had been based on weed control trials that lasted 90 days, and that no long-term soil biological studies had been conducted before the 1965 guidelines were published. She recommended that future herbicide guidelines include mandatory soil biological impact assessments before application rates are published. Poston's recommendations were implemented partially. By 1993, EPA pesticide registration protocols required soil microbial toxicity testing for new herbicide products, but the testing was not retroactively applied to products already on the market. Atrazine remained legal at rates up to 2.5 pounds per acre in most states, even though USDA had quietly reduced its recommended rate to 1.5 to 1.8 pounds per acre in 1987. The regulatory disconnect meant that farmers could still legally apply atrazine at rates known to damage soil biology. Many did, because the original guidelines had been in use for so long that the higher rates were considered standard practice in many regions. The practical impact of the 1987 rate reduction became visible in yield data over the following decade. Research published in 1998 by agronomists at the University of Illinois compared corn yields on fields where farmers had reduced atrazine application rates to 1.5 pounds per acre after 1987 versus fields where farmers continued using 2 to 2.5 pounds per acre. The study tracked 420 fields across 12 counties from 1988 to 1997. Fields treated at the lower rate showed average yields 8.7% higher than fields treated at the higher rate by the 5th year of the study period. Weed control efficacy was identical between the two groups. The yield difference was attributed entirely to improved soil biological activity. Earthworm populations on the lower rate fields averaged 134 individuals per square meter. On the higher rate fields, populations averaged 61 individuals per square meter. Nitrogen use efficiency, measured as bushels of corn produced per pound of applied nitrogen, was 17% higher on the lower rate fields. Soil organic matter increased on the lower rate fields and decreased on the higher rate fields. The data confirmed what researchers had been reporting since 1969. The USDA recommended rate from 1965 to 1987 had been damaging soil biology, and the damage had measurable economic consequences. The modern equivalent of the atrazine rate story is playing out with cover crop termination herbicides and soil health impacts. Glyphosate, 24D, and dicamba are used to terminate cover crops before planting cash crops. Application rates are set by manufacturers and endorsed by university extension services. Recent research has shown that repeated applications of glyphosate at labeled rates can suppress arbuscular mycorrhizal fungi and reduce nutrient availability in subsequent crops. The research is being published in soil biology journals. It is not yet reflected in application rate guidelines distributed by most extension services. The institutional delay between research findings and guideline revisions is shorter now than it was in the 1970s, but it still exists. The economic incentive to apply herbicides at higher rates for more reliable termination still conflicts with the long-term soil health incentive to use lower rates that preserve beneficial soil biology. What happened to Robert Henshaw is documented in county property records and USDA loan files. Henshaw continued farming his 640 acres near Goodland until 2003. He reduced his atrazine rate to 1.4 pounds per acre after his meeting with Carl Brennan in 1987. His wheat yields recovered over the following 4 years. By 1991, his yields were within 3% of county averages. He paid off his FHA loan in 1996. He sold the farm in 2003 for $420,000 and retired. Henshaw died in 2011 at the age of 73. His obituary in the Goodland Daily News made no mention of his role in prompting the 1987 USDA guideline revision. His family did not know the story until a Kansas State University researcher conducting an oral history project in 2009 interviewed him about changes in farming practices over his career. Henshaw told the researcher that he had done what any farmer would do. He had taken his records to the people who wrote the guidelines and asked them to explain why following the guidelines had cost him $14,000. He said he never received an apology. He said the revised guidelines appeared 3 months later with no explanation. He said he was glad the rate was lowered but that it should not have taken 22 years. The original 1965 USDA bulletin recommending 2 to 2.5 pounds per acre of atrazine is stored in the National Agricultural Library in Beltsville, Maryland. It is a 16-page pamphlet printed on cream-colored paper. The cover shows a photograph of a cornfield with no visible weeds. The recommended rate appears on page 9 in a table that lists application rates for 12 different herbicides. There is no mention of soil biology. There is no mention of microbial populations. There is no mention of earthworms. The guidelines are written as if the soil were a neutral substrate that held plants upright and delivered applied nutrients. The assumption was that killing weeds and feeding plants was sufficient. What happened in the soil itself did not require measurement. If your family farmed during the years when the original rate was in use, or if you applied atrazine at USDA recommended rates before 1987, leave a comment. Tell us where you were farming. Tell us what you noticed. These stories matter because they document how long it takes for institutions to correct recommendations based on incomplete testing. They matter because the same pattern plays out with new products every decade. They matter because someone needs to remember that 22 years is how long it took USDA to admit that the rate it had recommended was destroying the biological systems that make soil productive. Subscribe if you want more of these stories. They disappear when nobody writes them down. Institutional momentum that kept the high atrazine rate in place for 2 decades reveals something important about how agricultural science operates. It is not a system designed to quickly reverse course when evidence emerges. Once a recommendation is published by USDA and printed in extension bulletins and training manuals, it becomes embedded in agricultural practice. Farmers base their purchasing decisions on it. Equipment manufacturers design sprayers around the labeled rates. Chemical companies structure their sales forecasts based on it. Agricultural lenders evaluate farm finances assuming those yields will be achieved under those guidelines. The recommendation becomes infrastructure. Changing it requires admitting that the infrastructure was built on incomplete information. Most institutions resist that admission for as long as possible. The silence surrounding the 1987 rate revision is as significant as the revision itself. A major federal agency had recommended an application rate that damaged the biological foundation of soil health for 22 years. When that rate was finally lowered, there was no acknowledgement of the oversight. There was no explanation for farmers about why they should reduce their applications. There was no guidance on how to transition from the old rate to the new rate. There was no discussion of what farmers might expect from their soils in the years following the change. USDA simply published the new rate and moved forward. Farmers who had followed the original recommendations were left to figure out on their own that something had been wrong with the guidelines they had trusted. This pattern continues today in subtle ways. Extension services recommend application rates based on the best available science at the moment the recommendation is published. That science is often limited to short-term efficacy trials. Long-term soil health impacts are measured by researchers working independently, often in academic soil biology departments with limited funding and limited connection to the extension system that distributes practical recommendations. When the long-term research eventually contradicts the original recommendation, there is institutional reluctance to revise published guidelines because revision implies the previous guidance was incomplete. The solution is not to blame individual agronomists or USDA administrators. They work within the information available to them and within institutional structures they did not design. The solution is to change the institutional process itself. Mandatory soil biological impact assessments before herbicide rates are published would add time and cost to the registration process. But the cost of that assessment would be tiny compared to the cost of implementing a damaging recommendation across millions of acres for decades. Testing protocols could measure microbial activity, earthworm populations, and nutrient cycling rates over multiple growing seasons before application rates are finalized. Those tests could be funded by the pesticide manufacturers seeking registration, as they already fund efficacy and toxicity testing. The data would be public. Independent researchers could review it. Extension services would have robust biological impact data to accompany their application rate recommendations. None of this is technologically difficult. It is simply a matter of institutional priority and willingness to fund testing that might result in lower application rates and therefore lower sales for pesticide manufacturers. The deeper lesson from the atrazine story is that agricultural guidelines exist within an ecosystem of economic incentives that do not always align with long-term soil health. The chemistries work, the weed control is real, the short-term yield is there. What gets missed is what happens to the living systems in the soil when those chemistries persist at concentrations high enough to suppress beneficial organisms. Those organisms are largely invisible to farmers. Their work happens at scales too small to see, and in timeframes measured in years. It is easy to overlook them when the immediate need is to control weeds and maximize yield in the current growing season. But those invisible organisms are doing the work that makes yield possible year after year. When they are suppressed, their absence becomes visible only after several years have passed, and soil productivity has declined beyond what fertilizer applications can restore. By then, the damage is done. Robert Henshaw saw that damage on his farm in 1987 and had the clarity to recognize that following the guidelines had caused it. He had the courage to walk into a federal office and demand an explanation. He did not accept the answer that the guidelines were based on the best available science at the time. He asked why soil biological science had not been part of that assessment. He asked why 15 years of research contradicting the original rate had been ignored. He asked why farmers were expected to follow recommendations that had been proven incomplete. 3 months later, the guidelines changed. One farmer's refusal to accept an explanation is what finally prompted institutional action. If you are farming today and you notice something off about your soil, something that fertilizer does not fix, listen to that signal. It is probably telling you something about the biological health of your soil. Document it, measure it if you can, tell someone about it, share your observations with extension agents and soil scientists. These stories are how institutions eventually learn that their recommendations need revision. When you subscribe to this channel, you help ensure that these stories get documented and preserved. When you share them, you help ensure that other farmers do not spend 22 years following guidelines that damage their soil.